Well, hello there. Are you about ready for another exciting biology video? This one about passive cell transport. Well, you best be, because here it comes. All right, now before we start looking at how things get into and out of cells, we have to talk a little bit about the nature of matter in general. In junior high or middle school, you might have talked about something called the kinetic molecular theory. Well, if not, no biggie, because, hey, here it is. You know by now that all matter is made up of small particles. We call these particles atoms or molecules. But what you don't or might not realize is that these particles are in constant motion. So if you look at something that's solid, like a table or your desk or the floor, what looks like just something that's completely and totally stationary actually is made up of tiny atoms or molecules that are vibrating in place rapidly. Now they're held relatively close to each other so they don't fly away, but give them a little bit more energy. In other words, heat them up. Get that solid to melt. Turn it into a liquid. And then all of a sudden, the particles are allowed to move a little bit more freely. They'll slide past each other. Each particle will then have a little bit more energy. Well, give that liquid even more energy. Say heat it up. Get it to boil. You can turn that liquid into a gas of some sort. Now, the particles of a gas are going to be moving with a lot of energy, and they're going to be moving past each other, and they're not necessarily going to have to be in constant contact. They're essentially flying all over the place. So one of the things that we're going to have to realize here is that, hey, molecules are going to move all by themselves. We don't have to push them. We don't have to do anything. These things are moving. And that's going to explain a lot as to how stuff gets into or out of cells. All right, you've probably heard this before, but it doesn't hurt to do a little bit of review. We need to talk a little bit about solutions and solution chemistry. Recall that solution is just a special mixture in which one substance is going to be completely dissolved inside another. Now, the stuff that's dissolved is going to be the solute, right? And the substance that does the dissolving is the solvent. 99.99% of the time when we talk about stuff in our class, solvents in our class, we're going to be talking about water as being the solvent, right? So imagine taking some type of solute, whether it be, say, salt or sugar. Let's just pretend it's salt, and that would be our solute. We'll go ahead and stick that in solvent, stir it up, whatever. You eventually get it to all dissolve, and we have our salt solution. Now, we can talk about the strength of the solution and how much solute you have or how much solvent you have relative to each other. And we can talk about concentration. And chemists have a whole variety of different ways of describing concentration. We're going to make it easy and just look at percentages. So if we had, say, a 15% salt solution, right? That means by weight, it would be 15% salt. And flip side, 85% solvent or water. We're not going to get any more complicated with the math than using simple percentages. So we'll keep it simple. So what does all this have to do with cells? Well, imagine a cell as being just a container which is holding some type of solution. Dissolved wastes, nutrients, important cell products, whatever. And think of the outside of the cell as another solution. And we're going to see that there's going to be an interplay between the solution on the inside versus the solution on the outside. Essentially, that's what we're going to be looking at in something called passive transport. All right, we're going to be looking at passive transport first. Now, the concept of passive transport refers to the fact that in this case, the cell doesn't have to expend any energy, use any energy at all to get stuff into or out of it. Some substances will move across the membrane just because, well, molecules move. We learned that in kinetic molecular theory. But here's the thing. We want to look at this movement. This movement is not necessarily purposeful. It's random. But it has some definite effects. So imagine if you took a beaker of water and you put a little drop of dye in there. Originally, it might be pretty concentrated in one area. But because those dye particles are moving along with the water molecules, some of the dye particles are moving this way, some of them are moving that way, or that way, or that way, and all the way around, and eventually they're going to kind of sort of spread themselves out. What we're going to see is that the molecules will go from areas of high concentration to areas of lower concentration, so that the net effect is that everything is going to be all spread out, and eventually that whole beaker is going to be purple. So we call this process diffusion. Okay? So this is going to be an extremely important definition. The stuff in red you need to know. Matter of fact, if you know nothing more than the definition of diffusion and can apply this definition of diffusion, it's really hard to fail this unit. However, if you don't know this definition or you can't apply it, you're in big, big trouble. So commit this to memory. All right? Molecules are going to move from areas of high concentration to areas of low concentration. And, hey, this is not going to require any energy. Hey, a minor point, but an important one. Remember we said that after a while, the particles, the dye particles that we were looking at, are going to be completely and totally all spread out. So the concentration is going to be pretty much equal throughout the beaker. Now, it doesn't mean that the molecules stop moving. They're still moving. It's just, since it's random, just as many of the molecules are moving in this direction as in this direction. And probably just as many molecules are moving in this direction as in that direction. So the net effect is that it all stays more or less spread out. Now we want to go apply this idea of diffusion to something more than just, liquids in a beaker. Because we can also look at this in terms of movement in a cell. Mon ami, imagine this as a cell membrane. And yeah, I know it's missing some stuff, like some proteins, some integral proteins, some cholesterol and glycocalyx and stuff like that. But let's just look at the phospholipid layer here. The small red dots might be things like a solute molecule we're looking at. And if we looked at this as the inside of the cell and this as the outside of the cell, note that the outside of the cell right now has more of these solute particles than the inside. Well, assuming these solute particles could move across the cell membrane, they could go in or out, but since there's more of these particles on the outside, if the movement is truly random, you will eventually, just by chance alone, have more molecules going in than out. So we could think of diffusion as allowing molecules to move from areas of higher concentration to areas of lower concentration. And we're always going to go from high to low. Now this is sometimes referred to as a concentration gradient, or just a difference in concentration. Now, when we think of gradient, you might think of things like a slope. There's an easy way to remember this. If we were to have a basketball sitting on a ramp and you left that basketball unsupported, obviously it's going to roll downhill. You wouldn't have to push it. You wouldn't have to kick it. You wouldn't have to do anything to it. And this is a lot like what happens in diffusion. If this is our solute molecule, and you have higher concentration over here and lower concentration over here, it's going to have a net tendency, or these solute particles are going to have a net tendency to move from high concentration to low concentration, and you don't have to put any energy into it whatsoever. Now, assuming molecules can get across the cell membrane, diffusion is going to happen. However, it can happen at different rates. So things that affect the rate of diffusion or the speed of diffusion: concentration gradient, difference in concentration. The higher the concentration gradient or the concentration difference, the faster the diffusion rate. Again, think of our ramps. If our ramp is really, really steep and we release our basketball, it's going to go pretty dang fast. If our ramp is at a more gentle slope, that basketball is going to roll slower. So, same thing. If we were looking at a cell that maybe had a concentration of, say 15% dissolved solutes on this side of the membrane versus only 5% dissolved solutes here, we'd still be going from high concentration to low concentration. But the rate would be a little bit different than if we had something with, say, 25% dissolved solutes over here and only 2% dissolved solutes over here. The rate here would be much, much faster. Another thing that's going to affect diffusion rates is temperature. Now, temperature, to a chemist or a physicist, is just a measure of the kinetic energy that the substances, the molecules have, or how fast they're moving. So if you have higher temperatures, the molecules are going to be moving faster. And if they're moving faster, well, of course, they're going to get from point A to point B faster. So increase the temperature, hey, increase the diffusion rates. Decrease the temperature, make the diffusion rate slower. Other thing that's going to affect diffusion is pressure. Pressure can be thought of as a push given to molecules. And generally, molecules will move with a pressure gradient, in other words, from high pressure to low pressure. Certainly if you took a balloon and you squeezed the balloon here and here, right, whatever the balloon was filled with, air or water or whatever, would be pushed in this direction. Right? So, we're also going to have to look at pressure. Molecules will move from high pressure to low pressure. And the same rules apply with pressure gradients as you have with concentration gradients. The greater the pressure gradient, the faster those molecules are going to move. The other thing that might affect diffusion is how large or small the molecules actually are. Example, we had that cell membrane. We were trying to get these molecules to go across a membrane. Well, larger molecules tend to have a harder time getting through the membrane, and smaller molecules might have an easier time getting through the membrane. We'll talk more about size of molecules later. All right, a very important point. We said that diffusion will happen as long as molecules can get across a membrane. But remember that the purpose of a membrane is to control what goes in and out. So, turns out that properties of the membrane are going to restrict entry or exit of molecules. Think about how hard or easy it would be to get through a brick wall, or a chain link fence, or if you had an open window or a door in a wall. Membranes, turns out, can sometimes act like brick walls, and sometimes act like chain link fences, and sometimes act like open windows and doors. All right, now I know this puppy looks familiar because you've drawn that a bunch of times already, right? And again, function of a cell membrane is to control what goes in and out. If the membrane is completely permeable, that means anything can get in or out. But that's not the case of a cell membrane. Cell membranes are selectively permeable or semi-permeable. So only certain stuff gets in and out. Now, whether or not stuff gets in and out is usually dependent upon two things. One, in some membranes, not necessarily cell membranes, but maybe the membranes of certain organelles, you might have small pores, holes. And if the molecule is small enough, it's completely permeable to those molecules. If the molecule is too big, it's not going to fit through the hole, it's not going to get in or out. So sometimes that comes into play when we're looking at certain organelles. However, cell membrane itself, the membrane surrounding the entire cell, usually doesn't have large pores in it per se. Here, we're going to be looking more at the chemical nature of the molecules and their ability to dissolve across the membrane or squeeze past the chemicals that make up the membrane. So let's look at this a little bit more closely. Okay, since most of the cell membrane is made up of phospholipids, and forget about the proteins and everything else for just a sec, and those phospholipids have that kind of sort of fluid mosaic personality. If you have particles that are small enough, these particles, we said before, can kind of squeeze through either into or out of the cell. However, in order to squeeze through that phospholipid layer, you have to have molecules that are small, and they can't have any electric charge. They can't be an ion. Right? Gases like carbon dioxide and oxygen, hey, no problem. But if you do have a molecular charge, if you're an ion, right? Or even if you're highly polar, like water, right? You're not going to be able to squeeze through that layer. And if you're large, hey, forget it. You just can't make it. Now, that doesn't mean that you're stuck either inside or outside the cell. Some of these substances can still get into or out, but that's where those transmembrane proteins come in. So, yeah, now we got to look a little bit more closely at those puppies. If molecules cross the membrane with the aid of any of those integral proteins, we're talking about some form of facilitated diffusion. If you facilitate something, you make it easier. So the proteins are making it easier for these molecules to get across. Now note that this is still diffusion. You're still going from high concentration to low concentration, and you're still not requiring any energy to do it. The only rule is that we have to have some protein to act as a helper. So, turns out that there's two types of transmembranal proteins. One type, channel proteins. Channel proteins, basically you can think of them as having a little tunnel. But that tunnel has a particular shape, a particular form, and a particular chemical nature. So not just any old molecule can go through the tunnel. But if these molecules have the right shape and the right chemical nature, they can go through the tunnel no problem. Right? So here we have a situation where we have a higher concentration of these solute molecules over here, a lower concentration over here. Right? So diffusion can work. These will naturally go from higher concentration to lower concentration. Right? But they have to go through these channel proteins to do it. Another type of protein called carrier proteins. Carrier proteins, also transmembranal, but they work by actually changing shape to help bring molecules into or out of the cell. Again, we have to have a matchup. It's almost like an enzyme. Remember active sites in enzymes? Well, these carrier proteins have what are, in essence, active sites. If a molecule contacts the active site, the protein actually changes shape and releases the solute either into the cell or out of the cell. Still, whether you're talking about channel proteins or carrier proteins, you're talking about facilitated diffusion. You're always going from high to low. It doesn't require any energy. The proteins just do it as a part of their chemical nature. All right, one last very special note. Remember that water is polar, and because it's polar, it's not going to get across the phospholipid layer very easily. Remember, charged or polar molecules can't make it across that phospholipid layer, so you need transmembranal proteins to do it. In this case, we're going to need those channel proteins. And the channel proteins in this context are called aquaporins. Now, if the aquaporins aren't there, water can't get into the cell or out of the cell. However, here's the cool thing. All the cell membranes that we're going to look at have aquaporins. They have channel proteins that will allow water to enter or leave the cell. So if the function of the cell membrane is to control entry and exit, it is absolutely true that it depends upon the solute molecule you're talking about. Some solute molecules can get across the cell membrane, some can't. However, if you're talking about water, since cell membranes always have these aquaporins, water will always be able to enter or leave the cell. This is super critical. We need to remember that for our next lesson.